And now it's my pleasure to announce the next speaker, who I'm pretty sure is online. So that's Sophie Saunier. Sophie is Director of Research at INSERM, and she is the new head of the Laboratory of Hereditary Kidney Diseases at the Imaging Institute, following Corinne Antignac, who recently retired. And Sophie has a long-standing experience in kidney research, and she was involved in the identification of a number of genes that are causing nephronophthisis. And she is now working very hard in developing innovative approaches to therapies for nephronophthisis disorder. Okay, Sophie, can she start?
Hello, everyone. Would you hear me?
Hi, Sophie.
Yes.
Yeah, now we're ready to start.
Could you see my screen? Yeah. Okay. So thank you very much to inviting me today. Sorry that I cannot be with you in person. So yeah, as Franz said, our focus is mainly renal ciliopathy and in particular nephronophthisis. So as you hear before with Elizabeth, as renal ciliopathy are rare and severe genetic diseases caused by ciliary dysfunction. That is this specific organelles that is present at the surface of almost all cells, and the dysfunction of primary cilia will lead to many organ defects. And this is the main manifestation in renal ciliopathies is the kidney manifestation. And this includes a very known disease by you, either ADPKD or IRPKD, and nephronophthisis, as well as renal cystic dysplasia. So there is, unfortunately, it's end-stage renal kidney disease is the main cause of morbidity and mortality in the different ciliopathies. And there is today the only available standard care is dialysis and transplantation. So there is urgent need to find therapies for this kind of disease, but the difficulty is that there is this high clinical and genetic heterogeneity, and that limits the development of either diagnosis, prognostic, or efficient therapeutic approach. So 5 years ago, we start a French program called Silico for next generation medicine for renal ciliopathy at the Imaging Institute. And it's combined a group working on ciliopathy and also some data science and team that work with artificial intelligence, biostatistician, in order to improve patient diagnosis, patient classification, and in order to clusterize the patient according to a specific treatment. And in the parallel, we develop in our lab some specific disease model based on patient cells, either patient from the urines of the patient, the urex, or either iPS cells. I will come to that in a minute. With this, we will be able to study the pathogenicity of the disease and the mechanism, but also to identify some biomarkers that will allow us to decipher if there is a risk or not for kidney disease in these patients. And finally, the last approach of this program was to identify the new therapeutic targets. And what we did, and I will come to that in a minute, it's we choose the drug screen and a pharmaceutical approach to identify some specific treatment. So nephronophthisis is a rare disease. It's autosomal recessive chronic tubulointerstitial nephropathy. It's the first symptoms are polyuria and polydipsia in patients due to defect in urine concentration. It's generally, in contrast to PKD, AD, PKD, or PKD, the kidneys are small, not of normal size, and there is a dilatation, tubular dilatation at the late stage. But what is really typical of this disease is a massive interstitial fibrosis, as you can see here. There is a very early form that is more like IRPKD with a micro kyst in the cortical segment. So this is a major cause of end-stage renal disease during childhood and young adults. And as this is also a ciliopathy, the defect of the genes will cause also over systemic disease, and there is extrarenal symptoms, as you have seen with Bardet-Biedl syndromes in more than half of the patients. So mainly retinal defect, cerebellar defect, skeletal defect, or liver defect. So the characteristic of this disease, the nephronophthisis and related syndrome, is that it's huge. There is a large heterogeneity. So here we performed a molecular diagnosis on a large family cohort that we have in Necker, and we were able to identify allelic mutation in 60 patients in total. And as you can see here, there is half of the patient carrying mutation in the NPHP1 genes. NPHP1 genes encode protein that is localized at the base of the cilium, and together with the other partner that are also involved in the nephronophthisis, they are regulated the ciliary composition and the protein that are at the cilia. So this, you may see also that the other genes that are mutated in the disease encode also proteins that are part of the different modules of the cilia. So meaning that even if there is a large heterogeneity, maybe we can target the cilia and have the common defect rescued. So this is what we tried to do in the lab. So I will skip that. This is to show you also the repartition of the different mutations. And this is the models we used in the lab to identify the mechanism. For example, we used mouse models that recapitulate the nephronophthisis disease. And we identified by transcriptomic analysis the common transcripts that are altered. And Amandine Viau, a very talented researcher in my lab, she identified a specific cytokine signature in these NPHP models. But we are using other models I mentioned. We are also using urines from patients where we can collect tubular cells. And after immortalization, we can also analyze the transcriptome of these cells. And interestingly, we show that this signature in mice was also present in the patient with nephronophthisis. Also, what we did is we take directly the urine from the patient and we perform a single cell RNA-seq of urine from patient with nephronophthisis, but on also some age-matched LFT controls. And this is coherent because we saw an increase of immune cells in this urine that also suggest an immune response and cytokine, abnormal cytokine response. So finally, we validate this signature also in the kidney biopsy that we had at Necker, our collection. So there is definitely a renal inflammation in the nephronophthisis patient, and you can see that with all these models, we are able to follow this signature. Okay, so how can we target the cilia? Can it be an organelle to target to develop new therapeutics? So what we did is, first of all, we used, as Elizabeth mentioned, specific drug screen library that is composed of FDA-approved drugs. This is to fasten after the utilization of this drug if it's end up to be efficient. So we used as a readout some cells that were invalidated for NPHP1, that is the main genes, and we set up a cell-based assay based on two phenotypes, migration and ciliogenesis, meaning the capacity to grow cilia. So out of this screen, we identified 51 compounds that were able to rescue this phenotype. We select 11 of them that was pharmacologically different, and we use another model that I already mentioned, that is urine-derived retinal epithelial cells from our collection of URECs from patients. So these URECs that are, when they are primary cells, we immortalize them with thermosensible SV40 vectors in order to grow them in culture. And what we did is that they are thermosensible. We increase the temperature to make them differentiate and not cycle anymore. So with this, as we are really able to have a very nice readout of ciliogenesis. So we set up automatic quantification of ciliogenesis in these cells, and this was really a specific phenotype for NPHP1 UREC cells, that there were a huge decrease of ciliogenesis compared to control, age-matched control, but also to other CKD that are not ciliopathy. So we we validate our 11 hits that we previously identified in these UREC systems, and one of them ended up to be very, very efficient and to increase ciliogenesis, double ciliogenesis. This was PG1. What is PG1 or alprostadil? This is an analog of prostaglandin. Prostaglandin is known to be involved in pain, in regulating. It's a main prostaglandin expressed in the kidney, and it has several actions. And one of them is to target the 4 receptors, PGR1, 2, 3, 4, that are GPCR couple receptors. So when you look at the URECs, you see that the receptor that is mainly expressed there is the EP2 receptor. And this receptor, it's a GPCR couple protein to G-alpha-S, and it's lead to an increase of cyclic AMP. Interestingly, EP4, that is also expressed quite just after at the level of EP2, is also involved in cyclic AMP activation, and it has been shown to be associated to the ciliary membrane and to increase ciliogenesis via cyclic AMP. So this was a very, very good candidate. So we take out all of NPHP1 biocollection of URECs, and we test either PG1 that targets the 4 receptors or a specific agonist of EP2 receptors. And you can see that the efficiency was similar to both. So we check if the rescue was only at the formation of the cilia, or that there were also a rescuing of the composition of the cilia that is defective in NPHP1 patients. You see that in NPHP1 there is less adenylate CKS3 protein than in the control, and you can see that the PG1 treatment increased this intensity. So this was really a really good candidate. We validate the fact that this positive action was due to cyclic AMP increase. And it's what Alice did in the lab, a PhD student. She showed that there were after forskolin, that is known to activate cyclic AMP and PK pathway, there is this increase here, a similar increase with PG1 in NPHP1 cells. And so there is a mimicking activation of cyclic AMP PK, mimic the PG1 treatment. So what is known is that downstream the activation of cyclic AMP and PKA, there is this protein P27 that is known to induce the quiescence and induce ciliogenesis. So we prove that indeed there were this activation of this P27K1 after PG1 treatment. Also, another defect that is shown is an overactivation of Houillier in NPHP1 patients. And here again, downstream the activation of cyclic AMP PK, there were this downregulation of Houillier activation. So what that means is that maybe or probably there is really this efficient effect of this post-PG1 treatment in the whole phenotype in NPHP1 patient. So we performed some comparative transcriptomic analysis of NPHP1 in the urex of patients, and we identified genes that were inversely regulated in a patient treated or not. And what we saw is we showed that the enriched pathway was linked to actin cytoskeleton, production of extracellular matrix, and cyclic AMP signal. So this is, we further validate this. And as you can see here, in NPHP1 patient, there is this increase overexpression of laminin C2 here in EF. And you can see that this is reminiscence of extracellular matrix deposition. And when you treated cells with PG1, you can see that you decrease this production. So this is in vivo, this was in vivo validation that was really, really promising, because it's not only beneficially impact the ciliogenesis and ciliary composition, but the whole cytoskeleton and phenotype of the NPHP1 cells. So we went to the in vivo models, NPHP1 cow, and we generate a CRISPR cow with the deletion of the ATG in exon 1. And you can see that this model, there is no transcript and no protein either. So the phenotype of this mice was a mild infertility and very early onset of photoreceptor degeneration. Concerning the kidney, there were late onset renal lesion with tubular dilatation, abnormal cilia, and abnormal renal profiling. So we use these mice. We made a protocol where we treat the mice at 1 month until 5 months every day with intraperitoneal injection of PG1. And this was really, really successful, because we saw that decrease of the size and number of dilatation after 4 months treatment in the kidney of the male mice. What about the cilia? We also see, you see that in the NPHP1 cow mice, there is dilated tubules with elongated cilia. And you can see that with the treatment, there were normal cilia size again. So what about the pro-fibrotic markers? We show that by qPCR, there is an increase of some pro-fibrotic markers in the cow kidney mice. Here it's called 1A1. And with the treatment, there were a significant decrease. So we went to a global transcriptomic profiling of these mice, treated or not, and we can see that all the abnormal signaling pathway that were perturbed in the NPHP1 cow mice, naming like microtubule cytoskeleton, ECM organization, cytokine organization, actin organization, all what was really upregulated was going back to normal with the treatment. So globally, the PG1 treatment that was really non-toxic to this mice performance seems really to be beneficial for the kidney. So we use another model that was for the kidney, that is a zebrafish, that use also to have that have present with abnormal cyst in the pronephros, as you can see here, but that present also some abnormal distal dilatation and cilia length in the distal part of the pronephros. And what we show, it was that the alprostadil as a PG1 partially restore this severe cystic pronephric cysts, but also restore the cilia length in the distal part of the pronephros. So this is not NPHP1, it's another NPHP, it's NPHP4. So that means that suggests that other NPHP can also be eligible for this treatment. So here it's for the kidney, but what about the other organs that are also due to dysfunction of the cilia? I mentioned that the mice, as in the patient, has this retinal degeneration. Unfortunately, in mice, the retinal degeneration was really, really early and really severe. So we had a very, very short window to treat the animals. So, but we used, we didn't use a PG1, but we used an agonist of EP2 because we know that it's specifically expressed in the retina and that there is already an EP2 agonist that is used for treatment of the glaucoma. So we did an EP injection every 3 days from day 6 to day 21, because at day 21 there is a total degeneration, almost total degeneration of the photoreceptor. So what we show in this very short window, we show that there is an improvement of the outer nuclear layer of the mice treated with the agonist EP2. And this was also quantified by the electronic ERG, where you can see an improve also of the light sensitivity. So these results are, of course, it's not the best model, but this was really, really promising results. So what we show is that with this small molecule cell-based screen, we identified agonists that target cilia-related signaling pathway. This prostaglandin EP2 receptor agonist is able to rescue ciliogenesis in the patient cells, the UREX, the mechanism we have identified, but also it has an efficient effect on the mice models, on the renal defects, cilia defects, and fibrotic signature. For the EP2 agonist, we also show that the other tissue can be can be improved by the treatment with the partial rescue of the retinal degeneration. So now the question is the impact of this prostaglandin and Aβ2 agonist in UREC from patients with over mutation of NPH and over mice model. And this is ongoing in the lab, but also through a collaborative network that we have been setting up. So to finish, what's the next step of this therapeutic for renal ciliopathy? Now the limited action is that patients are really difficult to detect before reaching end-stage kidney disease. So it's really, really important to identify prognostic biomarkers. So it's really crucial to identify some predictive biomarker or prognostic biomarkers. So this is the next step. And what's going on? Where can we go for clinical trial? Where are we? So now we need a drug optimization. That is a company, Medesia, that is taking care of that. But for that we need really robust model for preclinical study. And for all, we also need to have a very good knowledge of natural history and all these patients and biomarker for treatment response. And how we will achieve that, this is through a new consortium and a new collaboration by extending the patient cohorts and the collaborator, that we will be able to gather enough patient information and enough knowledge and expertise to be able to bring these therapeutics to clinics. And this is what we did. We have this collaborative network that we set up. This is Therapies. It's composed of European clinician researcher, and that are really expert in the renal ciliopathies or kidney disease. There is also scientific and cell biology researcher, scientist. There is European patient advocacy, and there is also industrial that are dedicated to ciliopathies. So what we are doing, you can see the map here, and ERKNet is part also of the Therapies network. So the idea is really to integrate the main renal ciliopathy databases in Europe, and we will, for that, focus on two main pediatric renal ciliopathy diseases that are nephronophthisis, as I mentioned, and the related syndrome, but also ARPKD. So with this cohort and biorepository that are available in Europe in this consortium, and biocollection and patient samples, we will identify some really biomarkers that can really be predictive of the disease, and that can, in order to identify a patient that can be eligible for treatment. So you can see here that there is really expert and that people that develop some key models, such as, of course, specific mice models, but also some iPS kidney organoids, some tubuloids, to really decipher the robust models. What are the approach that we will use? So I mentioned the pharmacological approach with these very good candidates, but we have also the ISO oligo-antisense approach that Elisabeth mentioned just before, targeting the CEP290 gene, because John Saunier is also part of this consortium. So we can really try with this palette of therapies, try to target the bigger number of patients as possible. So the idea is really to cluster this patient with the clinical information, genetic information, biological information, using artificial intelligence method and data mining to cluster the patient according to the eligibility to the treatment. So this, of course, and I don't forget then, because this is the second part of the consortium, there will be also patients involved and patient advocacy and clinician to also improve and to have knowledge of report outcome measure that will be crucial also to set up clinical trial for this treatment. So with this consortium, we have a panel of all this expertise that we need. And with these promising results that are already present within the consortium in terms of models, robust models, in terms of drugs really available and ISO that are efficient, we hope to, in a few years, to be able to start a clinical trial for nephronophthisis on RPKD. So to finish, I would like to thank my collaborators. So here it's it's the French consortium that I present with the French also clinician and pediatrician that participate to this work. Medesia, that is industrial, that really accompany us for the development of the drugs, and all the Silico partners. But I would like also to thank my new partners, the European groups of Therapies that I'm sure will lead to a very successful, hopefully, collaboration. And thank you for your attention.
Thank you very much, Sophie. This was brilliant. You've taken us all the way from the cell-based compound screening to the clinical development of a new therapy for an inherited kidney disease. Thank you so much. Are there any questions in the audience? Yes.
Thank you very much, Dr. Saunier, for this amazing talk. I'm George Costa from Romania. I have a question regarding diagnosis of nephronophthisis, because I've seen quite a few patients with clinical pictures quite fully compatible with nephronophthisis. But when we had genetic testing done, we had only one heterozygous mutation. Do you think in the phenomenon of haploinsufficiency, do you think that that would be an answer? Or what's your take on that? Thank you.
Okay, yeah. So actually, when we did our genetic analysis, we identified heterozygous mutation, damaging heterozygous mutation in 20% of the cases. And usually now that there is at the genetic department in Necker, they have set up also the transcript analysis in order to identify the second event. So either doing a whole genome sequencing, but coupling also the transcript analysis, because most of the time we are able to identify the second event that is in the promoter region or entronic region. So there is two ways. Either you are missing the second event if you are not performing some whole genome sequencing, or transcriptome analysis, I mean, specific targeted transcript analysis, or either there is over genes, because there is also an enrichment of heterozygous mutation in ciliopathy genes in the ciliopathy cohort.
Thank you. And then we have a question online from one of our EPACs. She's asking, whether you have also patient associations represented in the new Terrasil project.
If I have patient inclusion, no.
Yes, a patient representation in this.
Yes. Yes, definitely. Of course, they take a main place. So I didn't. I mean, on the picture they were mentioning a Ciliopathy Alliance with Therapies. That is that was the main participants during the setup of this project. But of course, there is all the associations that are related to the renal ciliopathy.
Yes, and indeed, ERKNet, as you see, is also
the ERKNet, of course,
and there we will have direct interactions with this consortium. And there were three more questions. Okay, so I think Annette was first.
Yeah, thank you, Dr. Saunier, for this great talk. My name is Annette from Hannover Medical School. I have two questions. I was actually quite surprised to see your results on the immune infiltrate, and I wondered whether that was a secondary event or whether it actually did do anything to the progression of the disease. And then the second question would be, with the new treatment, did you see any effects on the immune composition in your model?
So I will answer the second one first, because I didn't hear the first one, but the second, yeah, definitely there were, for global transcriptomic analysis, there were this improvement of cytokine response, in the transcriptome analysis of the treated mice kidney. So now we are trying to test that more accurately, because we have specific cytokines that are activated in the NPHP patients. So we want to specifically look at this one, because it can also be response biomarkers for the treatment after. So yes, it seems to be the case. Your first question, sorry, I didn't.
The first question was whether the immune infiltrate had any driving force on the phenotype or the progression of the disease.
Actually, we don't know, but I think that it depends of it's what we are doing now. It's correlated the end-stage renal disease, I mean the GFR, with the cytokine production to see if there is some correlation and if it's, increase the severity of the disease. It's what we are doing now with all these biocollections of sample on our mice models. Yeah.
Thank you.
Then there's a question by Moin Salim.
Yeah, hi, Moin Salim from Bristol. Really exciting stuff. So my thought was, you mentioned the cyclic AMP pathway being affected, and clearly that's also affected in ADPKD and cell proliferation. So is there any crossover of your therapies for ADPKD?
Yeah. So, but you have seen that NPHP, either it's a ciliopathy like ADPKD, it's a completely different disease. And we don't have at all overexpression of cyclic AMP and proliferation. It's like when you have a loss of function of cilia reprotein, you don't have increased cyclic AMP. But when you have absence of defect of PKD and polycystins, you will have this increase of cyclic AMP. It's not what we observed, and we have less adenylate cyclase 3 present at the cilia. So we think that, and this is not only us, it's observed in several NPHP models that there is no cyclic AMP and PK pathway. It's more like a defective pathway. So this will, PG treatment will improve this aspect.
Mm-hmm. Yeah, if I understand correctly, it's actually the opposite
exactly
situation of what you see in ADPKD, right?
Yes, yeah, this is.
Okay, and then we have, okay. We have two more questions in the chat, and then we have to move on, I'm afraid.
Okay.
So Heidi is asking, you can see the question in the chat. Heidi, who's also a patient representative, is asking whether this model can be adapted for other rare kidney diseases.
Okay, so yeah, definitely. I mean, it's what we are checking. I mentioned a collection of UREX on the mice models where we have for other NPHP models on modules. But as we are also affecting some general downstream pathway of chronic kidney disease, we want also to check if this is compatible. And this has been shown that this EP2 agonist or EP4 agonist in nephrectomy, rat nephrectomy models and other models are improving. So I would think that it can have a larger action.
And the final question is, why can't we use this drug, which is already on the market right away in our patients?
Yeah, okay. Yes, why not? This actually, it's why I screen FDA approved library, right? To be able to move very fast into the clinics. But PG1 is not, it's degraded very easily, very rapidly. So now what we're doing with an EP2 agonist can be really more specific and more avoid also the side effect for long treatment. This is a chronic disease. So now, and it's more robust. I mean, it's stay more longer with low dose. So it's really what we are doing now with Medesia that is really putting a lot of effort to try to have the safest, the more specific molecule that is available. So.
Thank you very much again, Sophie.
You're welcome.
Great presentation.
Thank you.
Now it's my developments for hypomagnesemias.
So I can check. I'm sorry about it. I thought it can go this way. Yeah, I'm sorry. Yeah,
well, good morning, and thank you for the invitation and to give me the opportunity to talk about magnesium and to discuss how we could repurpose drugs to tackle diseases with hypomagnesemia. So I'm assuming that magnesium is not your favorite electrolyte, and okay, and that I need to give you a little bit of background regarding the topic. So the first question is, what is it? Yeah, okay. What maintains serum magnesium within a narrow range? And the answer is provided by very simple studies of magnesium intrusion in control subjects. And when you do this, you can plot how much magnesium is excreted in urine versus magnesium concentration in blood. And when you do this, you see that the normal range is this green box. It's possible to have no magnesium in urine with a blood magnesium that's still within normal range. And when you are in this situation, you cannot become hypomagnesemic. So the keeper of magnesium is the kidney. And it's a little bit too complex for me.
you can use these ones.
Okay, that's what I did.
Yeah.
Well, okay.
Yeah.
Thank you. And obviously the kidney is doing this by reabsorbing magnesium. So where is magnesium reabsorbed? And contrary to most electrolytes, magnesium is not reabsorbed a lot in the proximal tubule. only 20% about. Most magnesium is reabsorbed in the thick ascending limb of the loop of Henle, and about 10% is reabsorbed in the distal convoluted tube. So it has to cross the epithelium. There are two ways to cross an epithelium. You can cross the epithelium across the cell. This is the transepithelial reabsorption, and this is what's happening in the distal convoluted tubule. Or you can cross the epithelium between the cells, and this is the paracellular magnesium absorption that occurs in the thick ascending limb of the loop of Henle. So we'll be starting with that one and talking about diseases affecting paracellular magnesium reabsorption. I'm sorry, but I have to provide you with a cell model of electrolyte transport in the thick ascending limb. And you probably all know that the main function of the thick ascending limb is to reabsorb sodium chloride. It does this with this transporter NKCC2 that's targeted in Bartter disease. And by reabsorbing sodium chloride, this epithelium builds a transepithelial voltage that's lumen positive. And the lumen positive transepithelial voltage drives passively divalent cation, magnesium and calcium along the paracellular pathway. You can see shown on this picture that two major receptors are expressed and the basolateral membrane of this thick ascending limb cell. This is the type 1 PTH receptor and the calcium sensing receptor, and we'll be coming to this very soon. So the voltage that's the which drives magnesium reabsorption is mandatory. The other thing that's mandatory is the permeability of the paracellular pathway. And the permeability is due to the expression at the tight junction of very specific proteins named claudins. Claudins are part of a multimolecular complex expressed at the tight junctions. Claudins are a member of a family of 27 genes and more than 30 proteins, and they're expressed very specifically in tissues. They are all alike. They are small molecules with the amino and carboxytamyl ends within the cells, 4 transmembrane domains and 2 loops. The first loop bears the specific signature of each claudin, and the second loop and transmembrane regions are important for interaction between claudins expressed at the same tight junction. And actually, interaction between claudins seems to be important, and interaction can be done within same cell or between neighboring cells. This is homomeric interaction when one claudin interacts with itself, or it can be heteromeric interaction when one claudin interacts with another claudin expressed at the same tight junction. So we were able to show that in humans, shown here, as it is in the mouse and in the rats, in the thick ascending limb, we have at least 3 claudins that are expressed: claudin 10, claudin 16, and claudin 19. And what's something that is really specific is that the expression of those claudin is a mosaic expression. If you look at this picture, you can appreciate that some tight junction express Claudin 10. Some tight junction express Claudin 16, but no tight junction express at the same time Claudin 10 and Claudin 16. They exclude mutually each other. And which is not shown here is that tight junction that express Claudin 16 also express Claudin 19. They're expressed together. So we have two types of the tight junctions in the thick ascending limb, those that express Claudin 16 and Claudin 19, and those that express Claudin 10. And those are important for magnesium absorption, are the tight junction expressing Claudin 16 and Claudin 19. Actually, there is a rare disease that's named familial hypomagnesemia with hypocalciuria and nephrocalcinosis that is caused by pathogenic variants of Claudin 16 or Claudin 19 with very similar phenotype between Claudin 16 related and Claudin 19 related disease. They cause nephrocalcinosis that can easily be seen by ultrasonography. In this syndrome, there is no sodium chloride wasting. This is not Bartter syndrome, even it targets the thick ascending limb. But there is renal hypomagnesemia that can be extremely severe and hypocalciuria due to calcium wasting in urine because calcium and magnesium cannot be reabsorbed normally in the thick ascending limb, and we showed this 20 years ago. That thing, in addition to hypomagnesemia and hypocalciuria, is that the renal function usually degrades a long time in those patients, and a significant proportion of patients end in end-stage renal disease. We are able to study the Claudin 16 knockout mouse, and Claudin 16 knockout mouse actually have a decrease in the paracellular permeability of to calcium and magnesium when Claudin 16 is not expressed. Permeability to sodium and permeability to chloride are unchanged. How we did this? We used an old technique that's named microperfusion of isolated tubule. Basically, it allows you to work on native tissue and to dissect a tubule and to measure what can be reabsorbed by this tubule. And among things that can be measured, there are permeabilities. So the lack of Claudin 16 actually decreases permeability to calcium and magnesium. I told you that this segment expressed the PTH type 1 receptor, and we studied the signaling downstream the PTH receptor, and we showed using pharmacological tools that the signaling, the effect of the activation of the PTH receptor is gone when cyclic AMP or protein kinase A inhibitors are present, and also that the effect is gone when the calcium calmodulin kinase signaling pathway are added to the tubule. It also, on the other way, there is no evidence that the PKC-dependent pathway or ERK pathway are involved in the effect of parathyroid hormone in this segment. So we wanted to know what the parathyroid hormone does on calcium absorption, and this is normal tubules. And many people have shown this, that parathyroid hormone is able to increase calcium absorption on isolated tubule from the thick ascending limb in the normal mice. But we did the same when Claudin 16 is gone. And we were not expecting this. It shows that parathyroid hormone still increases calcium reabsorption, even if Claudin 16 is not there. We did the same with magnesium, and the result is pretty much the same, although this group is not completed. Parathyroid hormone increases magnesium reabsorption in Claudin 16 expressing mice, but also in Claudin 16 knockout mice. And we targeted the other receptor expressed at the basolateral membrane, the calcium sensing receptor, with calcium sensing receptor antagonists. And in the Claudin 16 normal mice, the calcium sensing receptor antagonism increases calcium absorption, which is expected. And the same is true when the Claudin 16 is gone. So despite the lack of Claudin 16, it's possible to increase calcium and probably magnesium reabsorption in the thick ascending limb. And this is the basis to what I will be talking in a few minutes. So if we move a little bit downstream, we're in the distal convoluted tubule, and there magnesium is reabsorbed transcellularly. And there are many diseases affecting magnesium reabsorption in the distal convoluted tubule, including many genetic diseases. Magnesium absorption can be impaired because of the lack or abnormality of the magnesium channel TRPM6. It can be indirectly impaired because of the lack of function of the sodium chloride cotransporter NCC, or it can be impaired because the exit of magnesium out of the cell is impaired, or it can be impaired because this transcription factor HNF1 beta is abnormal and cannot maintain the transcellular transport of magnesium across this cell. Incidentally, many studies studying the effect of SGLT2 inhibitors in mostly diabetic patients have reported that SGLT2 inhibitors are associated with an increase in plasma magnesium reabsorption. There have been several meta-analyses showing a significant effect of SGLT2 inhibitors. The only study I will show you today is that one, reporting the effect of SGLT2 inhibitors in 3 patients, including 2 patients with NHF1 beta deletion. And what is shown is the magnesium concentration expressed in milligram per deciliter without or with treatment with SGLT2 inhibitors. And you can appreciate that in all cases, this is not controlled. But in all cases, magnesium concentration in blood is higher when SGLT2 inhibitors is given to the patient than when SGLT2 inhibitors is not given to the patient. And the increase is quite significant regarding magnesium concentration and with what we can obtain with, let's say, conventional treatment, which is just giving magnesium orally. So based on this, I've been asked to coordinate a work package on new treatment for tuberculosis, and this is part of proposal given by a consortium. So we're in competition with the first one shown by Sophie. And the name of our consortium is ERKNet. The goal is absolutely the same. It is targeting rare diseases. So we'll see at the end. And as I told you, one work package is on tuberculosis. So what we propose is very simple. It's based on preclinical and clinical data, is to conduct pilot studies in patients with specific renal diseases, either patients with diseases in transcellular renal magnesium transport due to NHF1 beta, trypan6, CNNM2, or NCC abnormalities. And the drug that will be tested will obviously be SGLT2 inhibitors. And another pilot study will be on patients with defect in paracellular magnesium and calcium transport due to Claudin 16 or Claudin 19 abnormalities in FHHNC, and drugs that will be studied will be the calcium sensing receptor antagonist and teriparatide in order to target both receptors expressed at the basolateral membrane. If I may, I would just like to end by another story that's not included in the proposal, but that illustrate that innovative and specific therapy can be driven by the discovery of a specific cause. With Fadi, who is in this room and speakers this afternoon, we could take care of a patient, 70 years old, who has been admitted to the hospital because he had muscle weakness, paresthesias, cramps, and muscle twitching. He had almost no history except autocoronary bypass and mitral annuloplasty. And at that time, magnesium and calcium in blood were perfectly normal. He had no diuretic therapy, and this is the clinical or biological picture of the patient. Extremely low magnesium, extremely low calcium, a lot of magnesium in urine, a lot of calcium in urine, renin perfectly normal, aldosterone perfectly normal. So this is a syndrome of isolated renal magnesium and calcium wasting. This is FHHNC, obviously. 70 years old, Claudin 16 gene perfectly normal, Claudin 19 gene perfectly normal. So we made the hypothesis that this is acquired FHHNC due to autoantibodies against one of the two claudins expressed in the thickest ascending limb. And actually, we showed that the plasma of the patient was able to label specific structures within the thick ascending limb, and you can appreciate that the pictures have a pattern of tight junction. And to make a short story, long story short, we are able to show that the target was Claudin 16, not Claudin 19. And so it was hypomagnesemia, hypocalcemia, and interstitial nephropathy. Am I too long? Caused by Claudin 16 autoantibodies, and Fadi, I must say we have to use rituximab and plasmid changes. So we did it. Not quite successful on magnesium, but we were more successful on calcium. Calcium could be more or less maintained without any calcium supplements. And we were able to improve renal function, at least for a while. This therapy was specifically targeted for this patient because we were able to show that it was due to autoantibodies. Probably I've forgotten many people who should be on this slide. But I thank all people I've been working with in the last years. I would like to add all participants of our proposal ERKNet who are here and online. Thank you.
Thank you very much, Pascal. So the SGLT2 inhibitors seem really to be miracle drugs. They work for everything, even hypomagnesemia. I see two questions. The first one in the back.
Hi, Pascal, fantastic talk. I'm Francesco Trebiccione from Naples.
Yes, I know you.
But I'm really fascinating of this rule of SGLT2 because we also know that they are associated with increased potassium excretion and not hypocalcemia, but decreased potassium level. How do you think will be the molecular mechanism that couple magnesium increase and potassium decrease? They usually go,
This is part of the proposal.
Yeah. I would like to know your
Unraveling the mechanism is part of the proposal. So it could be many things, actually. It could be direct or indirect. When you do something that is as brutal as preventing a lot of glucose reabsorption in the proximal tubule, what can be done downstream is mostly unpredictable. Because those cells in the distal nephron have many sensors, but they can be affected by what's flowing, and that normally does not flow in urine. But they have also many receptors that obviously can be affected by the change in mineralocorticoid activity or what you can imagine.
Thank you. And Roxana.
Thank you very much. Magnesium is thought to have a protective role against vascular calcification in clinical studies, and maybe SGLT2 inhibitors will help with that as well. But what is the mechanism of magnesium entry into the smooth muscle cells and its action?
I don't know how magnesium enters into smooth muscle cells. I should.
That might be interesting because there's a lot of attention on magnesium, even magnesium-based binders, phosphate binders, because of its protective role on calcification.
Well, magnesium entry into cells is usually easy because of the cell membrane voltage. Magnesium in the divalent cation. It's about the same within the cell and concentration. Outside the cell, within the cell, there is no real chemical gradients. But because of the voltage, magnesium can flow into the cell if you give it a channel.
And can it competitively bind against calcium?
I don't know. Oh, no, usually magnesium channels are somewhat specific, not like cloudins, but share properties.
Could you consider this in your proposal? Or?
It's too late.
Next time.
Okay, so we have a few more questions online. Sharon Devay is asking what is the mechanism for the PTH effect on calcium transport in Claudin-16 knockout mice?
I would be happy you give me the answer. We're actively looking for the answer. We don't know. But this is, I agree, this is really intriguing. What's going on in this? So you can imagine this is Claudin-19, but the expression of Claudin-19 at the tight junction with when Claudin-16 is done is not normal either. So it's probably not that. We currently are not able to explain what's happening. We see it's happening.
Mm-hmm. And then we have a comment that FHHNC patients have often quite normal magnesium levels. Whether you know what fraction of patients has relevant hypomagnesemia, and if there is a relevant amount that has low magnesium, what is the clinical endpoint for possible studies? The delay of CKD?
I would disagree with the assumption that most patients with FHHNC have normal magnesium. They can have normal magnesium when they have CKD, because obviously CKD decreases loss of magnesium. But when they are younger, most of them have hypomagnesemia. So yes, one endpoint would be targeting CKD. But before this, we need to be able to show that we can do something on the short term on the ability of the thick ascending limb to change its phenotype. And if yes, we can base on this to propose longer time studies and to see what happens to CKD and if we can prevent it.
Mm-hmm. And a related question is whether one could tolerate higher PTH values, given what you've shown, in patients with FHHNC.
If we can tolerate
higher PTH levels.
It depends on what we call higher. This is actually one of the purposes, is to determine how much PTH we can tolerate.
Yeah. Okay. Then we have comments also from patient organizations for hypomagnesemia that say that they will be very happy to help finding patients for the clinical trials.
Thank you very much.
Yeah, and this again, since ERKNet is participating in this trial application, it would also be us that could link you to the patient organizations.
Thank you.
Okay, very good. So thanks again, Pascal, and we will move on to the